Okay, apologies for the long delay and apologies for my being a little bit haggard today, but I've been working hard. It's something I think all of you will be very interested in and relating to this issue of milestones towards a cure. We received RFP, a request for proposals, from the Multiple Myeloma Research Foundation, MMRF, here in the U.S., headed by Kathy Giusti, Guisti, I believe. Please Google her if you're not familiar with her work. She's a phenomenal individual and very ambitious, and her goal is to use the energy and the discipline of the private sector of the business world in academia to try to accelerate cures and progress in multiple myeloma. So it was a very ambitious proposal framed around a clinical trial, which is going to be 1,000 patients with newly diagnosed multiple myeloma. And the milestones here, the milestones that they're very interested in are from these 1,000 patients. Number one, how can we tell who's going to respond to treatment and which treatments? And how can we tell when someone has minimal residual disease? We know now that we can have apparently complete responses in multiple myeloma with these new drugs, but we also know that people relapse after those dramatic responses. In CML, we have a molecular test. We can have a molecular remission. We don't have that in multiple myeloma, so we're going to need to develop that. Nowhere else in the world, I think, from WashU can put together the expertise needed to respond to this, because what they requested was to biobank thousands of samples from these patients and to analyze them using state-of-the-art molecular tests, many different types of molecular tests. So I'll tell you briefly what we did. We have Sashi Kulkarni here is our FISH and cytogenetics clinical genetics expert. So we're going to do traditional testing as well as SNP arrays, single nucleotide polymorphism arrays in a CLIA certified setting. That means it's a hospital environment so that the results can be reported out. Kathy Weilbacher is brought on board because we're not just going to be looking at the myeloma cells. She's an expert in the stroma, and we want to look at the myeloma cells, the CD138 cells, as well as cancer-associated fibroblasts, the T cells, and early B cells that may have clues to these questions. Sandra McDonald here is going to be the banking and pathology, is going to be banking these samples. It's a huge endeavor with the help of Rakesh Nagarajan. So these are sub-milestones basically, as we need to keep track. We need a database that can keep track of not only regular clinical tests like FISH, cytogenetics, but if we get sequencing and proteomic data, novel tests, we need a database and informatics to keep track of that. Rakesh is a world expert in bioinformatics and is going to put together a database to keep track of this. We have on board Bob Fulton, who's I like to think of him as Scotty from the Star Trek. He's the engineer, the man who makes the engines work at the Genome Institute at WashU. The genome sequencing effort is a phenomenal world-class enterprise, I think, on par. The only comparable institution would be the Sanger in Britain, with a focus here on whole genome sequencing. But then beyond whole genome sequencing, we're going to do methylation status, because we think maybe not just DNA, but epigenetic events are going to be important, as well as RNA-seq. And this gets a little technical, but not just sequencing the DNA, but the RNA as well, we think is going to be very informative. And then we have Reed Townsend, who's proteomics. We need another sub-milestone, as we need to understand not only in the serum, but in the bone marrow plasma, what protein biomarkers are there that may correlate with response to therapy or minimal residual disease. So we can have a test in the clinic to figure this out. And of course, Ravi Vij, our master myeloma clinician, who established a relationship with the MMRF and has been working with them quite well. We could not have gotten this opportunity without him. He'll be in charge of supervising the biobanking side of the effort, and I will be in charge of overseeing the scientific part of the effort and attempting with the MMRF to impose a sort of market discipline on this otherwise academic effort to make sure we hit milestones and make rapid progress in the 8-year span of this trial. So this is a huge effort. We're thinking of going out to Connecticut to pitch this huge proposal. It'll be a big deal if we get it. Even if we don't, it's been a great exercise, and now you know framework number one for myeloma. There's another framework of milestones I'm developing for MGUS and smoldering myeloma, an early phase, and that's a critical part of this. But for myeloma, that's the plan.